Welcome friends, we are going to start a new lecture series on the topic of proteins, and in this course we are going to discuss about the introduction to proteins, then the protein building, that is how the proteins are built using the building blocks, that is the amino acids. Then we will move to the classification of amino acid, that is the division of the amino acid, and then the ionization, how these amino acids are ionized when we put them into the solution, and then the pKa value of the amino acid. This is very important. And then finally the structures of the proteins, and there are basically four types of structures: the primary structure, secondary structure, and tertiary, and the fourth one is the quaternary structure. In this video we are going to discuss about the introductory part of the proteins. So what are proteins? We start with these questions. These are the largest macromolecules. Now what are macromolecules? These are simply the biomolecules. We know there are four types of biomolecules. The first one is the carbohydrates, okay. The second is the lipids. The third one is the nucleic acids, and the fourth one is the proteins. Let me use another marker. This is not dark. Personally, I feel these proteins are the most important of all these biomolecules. We are going to know why they are so significant. I must say, all the other fields of biology, more importantly the biochemistry, they are somehow related to proteins. Or I can say, at the end, listen to this statement, at the end, these are the proteins which are actually executing the function. Or we can say, the functions of these biomolecules, that is carbohydrates, lipids, and nucleic acids, they are somehow dependent on the functions of the proteins. Or we can say, these biomolecules, they are performing their functions in conjunction with these proteins. We want to justify this statement. To understand this statement, we relate the concept of proteins to molecular biology. We know what is molecular biology, in which we study about the replication, transcription, and translation. And what is this? This is called as central dogma of biology. That is, we have DNA, okay, a copy of DNA which is doubled or copied to another DNA molecule, and this is known as replication. This is the first step. After this, the DNA is transcribed into RNA, and this is known as transcription, okay, transcription. After the RNA is formed, this is converted into what? I will tell you later, okay. This process is known as translation, and the product which is formed here is the proteins. That is what we are talking about, okay. So the proteins are formed at the end, and if we talk about this replication, where one copy of DNA is formed from the previous or existing copy, the process is carried out by certain molecules which are known as helicases, and the replicating enzymes, the DNA polymerase. These enzymes and many more enzymes, they are actually proteins. We know the enzymes are proteins. So the DNA replication is using enzymes as proteins to carry out the completion of the DNA replication. And now let's talk little bit about the cell biology and its link with the proteins. We know what is cell biology? Biology of the cell. The processes which are occurring in the cell. Suppose this is the cell. There is inside and there is outside of the cell. This inside is known as cytoplasm. Outside portion is the extracellular matrix or exoplasm simply. To carry out a function within a cell or the process inside of the cell, we need a signal which must be coming from the outside of the cell. Let's say here. Let's take an example here. Suppose that signal producing molecule is the EGF, which is present outside of the cell. This EGF is basically the epidermal growth factor. Okay, this needs to bind to a molecule which is present here on the periphery of the cell. This is known as EGFR, that is epidermal growth factor receptor to EGF. Okay, so these molecules, they are nothing but proteins. Okay, and what they do is that they constitute a signal transduction pathway in which one protein is binding to a receptor, we can say, and this binding results in the activation of a downward protein which is present, you can say, here in the cell, and after its activation it results in the signaling pathway. Or the signaling cascade is initiated. Now we can take one more example from cell biology of cell cycle. What is cell cycle? A process in which daughter cells are produced. This process or the cycle is comprised of few stages or phases which are known as the G0 phase, S, G1, G2 and M phase. These phases or these stages, they are regulated by regulating machinery which is known as checkpoints. Also there are cyclins plus cyclin dependent kinases. These are nothing but proteins. A checkpoint is a regulating protein which stops, hinders or simply does not allow the progression of one stage to the other stage unless and until this checkpoint protein is not sure that the previous stage has been completed or not. Okay, and in this way this is making it sure that the cell is not committing any error. Okay, and if these proteins are misbehaving, what could happen? Two things. First is overgrowth. The second one, no growth or retarded growth, you can say. This would lead to cancer. In both cases, the cell has to experience negative consequences which the cell does not want to have. Now if we talk about the metabolism of carbohydrates, these sugars, they also employ proteins to convert one form of carbohydrate to another. Let's take an example of glycolysis. This is enzyme catalyzed process in which glucose is converted into pyruvate. This is 10 enzyme catalyzed process where first glucose is converted into glucose 6-phosphate and then fructose 6-phosphate and so on, finally to pyruvate. So 10 enzymes are being used here, and enzymes are nothing but proteins, and if these proteins, they are not present, what would happen? The glucose cannot be converted to pyruvate and hence no energy. Now proteins as immunological point of view. What do we study in immunology? Immunology, whatever you say, immunology. The study of the immune system, basically the antibodies. Now what are antibodies? the proteins which bind to the foreign particles such as the viruses and the bacteria, so that the body can be protected from the infection causing microorganisms. Now, apart from these functions, these proteins, they also play other important roles such as the storage and transport of certain metal ions. Storage proteins, they store certain metal ions such as the iron. There is a protein which stores or which in which the iron is found is known as ferritin. Ferritin, which stores iron in it. Okay, and this iron is a component of heme. Okay, which is found in hemoglobin, and hemoglobin is a protein which transports oxygen. Now, by giving these examples, what I am trying to say is that whatever area of the biology, whether it is microbiology, it is immunology, it is biochemistry, or it is molecular biology, you must have the basic understanding of these proteins, because you are going to notice these proteins in all of them.